So if you watched a previous video, we learned about the four important organic molecules, and we learned how they're built from monomers that are connected to form polymers, single units connected in a chain to form polymers. And there's four main types of organic molecules: carbohydrates, lipids, proteins, and nucleic acids. In the previous lecture, we learned about proteins. We know that proteins are made up of amino acid monomers joined together to form long chains. In this video, we're going to be talking about carbohydrates, lipids, and nucleic acids. And we're going to talk about two main things. The first is we're going to talk about the structure of each of these molecules. So, what are the atoms that make them up? What do they look like? How are longer polymers made from individual monomers? What kind of bonds are they made up of? Just basically everything you need to know about their shape. And this is connected to your You Must Knows, talking about the four important macromolecules or biomolecules, or another word is organic molecules. So make sure you're thinking about the structure and the function. And what would happen if you were to change up parts of the structure? How would that affect the function of these molecules? Function, remember, means its job, what it does in the body. So just to recap really quick, we know that there are four types, and each one is built up of smaller pieces, so kind of like the building blocks. So we have four types of macromolecules: nucleic acids, proteins, fats, and sugars, or polysaccharides, and the individual pieces. So small sugars make up larger chains called polysaccharides. Another word for that is carbohydrates. Fatty acids are the small pieces that come together to build fats. Amino acids are the small monomers that are joined together to build proteins, and the nucleotides are small units that join together to form nucleic acids. So as we go through this video, I want you to think about, I have a little graphic organizer somewhere in here. Here we go. Okay, if you have a piece of paper, this will really help you, or put this in your notes somewhere. I want you to, as we're going through the four molecules, so protein, carbs, nucleic acids, and lipids, draw a picture of what the monomer looks like, what its structure looks like, and then write some examples of polymers with their functions. So for example, in this video, you're going to learn about carbohydrates. So write the name of the molecule, write the name of the monomer. So sugars, fructose, draw a picture of its structure, and then write what would happen if you were to link up many of these monomers, polymer, and some examples of their functions. And so do that for carbs, lipids, proteins, and nucleic acids. And I promise you, this will really, really, really help you, because there's a lot of words, there's a lot of pieces. You're going to need to keep this organized. So take out a piece of paper, do it right now, even if you're watching this in the car. Trust me, this will pay off a lot later when you're trying to memorize all of this. There's a lot of details. And we can go over this too as a class. So do that, watch the video, and you'll be great. So our next macromolecule that we're going to be discussing, found in living organisms, are carbohydrates. And just a fun fact, carbohydrates is a fancy word for sugar. We're going to learn that carbohydrates are made up of sugar. That's the monomer of a carbohydrate. And Americans consume an average of 140 pounds of sugar per person per year, which is an interesting fact. So carbohydrates are composed of the elements carbon, hydrogen, oxygen. That's why it's called carbo for carbon, hydro for the hydrogen 8. And they're always found in this ratio of elements. So one carbon to 2 hydrogens to 1 oxygen. So you can think of it in this formula. So if you have one carbon, there's always going to be 2 hydrogens to that carbon, and then 1 oxygen. So let's say, and then if you have a multiple of that, so let's say you multiply that by 3, then you would have, or 6, then you would have 6 carbons, 12 hydrogens, 6 oxygens, C6H12O6. And so whenever you're talking about sugars, it's always going to, if you counted up all the carbons, hydrogens, and oxygens that make up that molecule, you would find that ratio. When we're talking about macromolecules, we want to think about, number one, the structure, what it's made up of, the atoms that it's made up of, and how it's bonded together, and also the function of the macromolecule. So for carbohydrates, the main function is energy. To get energy, quick energy, when you're eating carbs, you're going to get energy from those carbs. Storage of energy, so sometimes you don't need to use energy right away. You can store it for later, for when you run out of energy. Raw materials and structural materials. So what this means is carbohydrates can help build up the structure of your cells. The monomer, the building blocks, the bricks of carbohydrates are sugars. So if you take one individual piece, that's called a sugar. You put multiple together, it creates a long polymer. An example of a carbohydrate are sugars and starches. So if you took a tiny sugar molecule and put it in a long chain, it would either form a sugar or a starch. There's a bunch of different types. When you're thinking about sugars, just a little quick thing, when you're naming them, they often end in ose. So if you find a molecule that ends in ose, it's probably a sugar. And the first part of the name tells you kind of how many carbons are in the molecule. So hexose would be 6 carbons. Another word for that is glucose. We'll look at glucose in a second. There's 5 carbons. Pent means 5. Ose means sugar, so 5 carbon sugar. That's fructose and ribose. Triose is glyceraldehyde. So there's 3 carbons. That's a 3 carbon sugar. So let's talk about, well, what does a sugar look like? Well, right here, this is a basic sugar molecule. And what we'll see here is that scientists get lazy when they're drawing carbons and when they're drawing sugar molecules. Oftentimes you'll just see that, but what it really means underneath is at each of these vertices of this shape here, it stands for a carbon. And remember, carbon forms more than just 2 bonds. Right here, it's just showing 1, 2 bonds. There's actually hydrogens that are hiding here. So there's a hydrogen branching off. So if I were to draw it here, if you really added, you would see a hydrogen and a hydrogen here. And then same thing all around. Hydrogen branch here, hydrogen, 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 hydrogen. And so for this molecule, we have 1, 2, 3, 4, 5, 6 carbons. So C6. And then let's remember our formula. So there's 2 hydrogens for every carbon. 6 times 2 is 12. And then O, 6. Now again, there's actually a bunch of oxygens surrounding this too, but scientists get lazy and they really just draw that when they're drawing sugars. Now we're going to be numbering our carbons. So we assign numbers to each of them. Now we start with the carbon right next to the oxygen. So on the right side of the oxygen, that's carbon 1. And then we go around in a circle. Carbon 2, carbon 3, carbon 4, carbon 5. And then we have this extra carbon. It's kind of like the chimney on top. That's carbon 6. All right, so there's different ways to represent this brick here. So this right here is 1 sugar. And this is like the detailed version. So if you were to draw all the carbons, hydrogens, and oxygens, this is actually what it would look like. So here's our oxygen that's in the ring. So then to the right of it, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6 is a little chimney. This is C6H12O6 like we just looked at. But scientists like to get lazy and just draw it like this. Now this right here, this is called glucose. So this monomer of carbohydrates is kind of like the brick that builds up the entire chain of a carbohydrate. So like we just talked about, here is our glucose. And you can see it has 6 carbons, 1, 2, 3, 4, 5, 6. And notice that it's bonded here covalently to a new molecule called fructose. What is fructose? Well, notice it's kind of structured similarly to glucose, but here it has less carbons. So 1, 2, 3, 4, 5 carbons. And then you have one here on the end that will be bonded to another one eventually. So this one will eventually just have 1, 2, 3, 4, 5 carbons. And so when a glucose and a fructose, these are 2 different types of monomers, are bonded together, it forms a whole new molecule called a sucrose molecule. Now, this is what you find in your sugar when you are adding sugar to your coffee. This is table sugar. It's that white powdery sugar that you find in your cabinet. And that's when you take a glucose monomer and a fructose monomer, join them together to form a polymer. Now, if you take one monomer and another monomer and join them together, we call that a disaccharide. So saccharide means sugar. One of them by itself, we call this a monosaccharide. Mono means 1, saccharide means sugar. So 2 bonded together are called disaccharide. And you can kind of mix and match what kinds of monosaccharides you want to add together to make a disaccharide. So here we have sucrose. So sucrose, on the left we have a glucose molecule bonded to it. It's a fructose molecule. When you put those 2 together, you make sucrose. Lactose, right here we have galactose bonded to glucose, makes lactose. Here we have maltose, so glucose and glucose bonded together make maltose. Now, this is in the food that you eat. So sucrose in your sugar, lactose is in your milk. And what your body does is it's actually going to cut up these sugars into smaller pieces. So it's going to break this bond here and break up the individual monomers into small pieces. And that's called digestion. Digestion is breaking up larger molecules into smaller pieces. And then your body uses those to get energy. So we have different types of sugars. So again, like we just talked about, monosaccharides means one sugar. And so that's a simple monomer. It's one single unit. So just like this is a monosaccharide. And then example would be glucose. Disaccharides is when you take two. So you could take a glucose and a glucose, or a fructose and a glucose, and you bond them together. Di means two. So disaccharide is two monomers bonded together. An example would be sucrose. And then a polysaccharide, poly means many. If you were to take many monosaccharides and bond them together on a chain, you would get a large polymer, a really long molecule. And examples of polysaccharides are starch, and we'll talk about some others as well. So basic types of carbohydrates, glucose is really small, sucrose is a little bit bigger, and then starch is a really long polysaccharide. Okay, so how do you take a monosaccharide and then build a larger one? Well, that happens through a process called dehydration synthesis, which we talked about in our previous lecture. Remember, if you just have two monomers hanging out, they're not going to want to bond together. In order to bond two monomers together, you have to remove something. And in this case, we have our OH group here and our OH group here. And if you take off the OH and the H, you're going to form water. And then these two molecules are going to form together. They'll link up and form a bond. So this is now our new covalent bond. We call it glycosidic linkage, but that's really just a covalent bond. So dehydration synthesis, because you're dehydrating, you're removing water, and you're forming a bond between these two monomers. Now you go from two monosaccharides to a disaccharide, and they're bonded together. So glucose and glucose now makes maltose, which is in your food. Another example, so if we took a glucose and we did dehydration synthesis, synthesis means to put together, they would form sucrose. So this is your table sugar, disaccharide. Now, disaccharides, they're in your food, but you can also build really, really long sugars called polysaccharides. Now, polysaccharides, some things you need to know about them. Number one, they're not soluble in water. So if you put a polysaccharide in water, water would not really be able to break it up or dissolve it. They're also too large to pass through cell membranes, so they can't get into your cells. So if you eat a polysaccharide, you can't get it into your cell. You need to get it into your cell because your cell will chop up the polysaccharide into smaller sugars, smaller pieces, so that you can get energy from that molecule. And so we're going to look a little bit about how that happens. So two main functions of polysaccharides. Number one, they help your body store energy. Number two, they give your cells structure. So they kind of hold your cells together. So let's start with energy storage. And we'll start with plants. Plants are living organisms. Now, when plants need food, they go through a process. Think in your head, what is the process that plants do to make their own food? Hopefully you said photosynthesis. And photosynthesis is the creation of, or the synthesis of a sugar molecule called glucose from carbon dioxide from the atmosphere and light and water, all mixes together and makes this beautiful glucose molecule. That's where we get the beginning basic building block of all sugars, is plants literally make it. But the plants are going to eventually use that glucose molecule for energy, but sometimes they don't need it right away, so they want to store energy. And what they do is they take individual glucose molecules and they'll kind of put them away in the closet, and they'll do that by bonding together a bunch of glucose molecules on a string, and that's called starch. So we have one glucose monomer bonded to another glucose monomer, bonded to another, and it will form a really long polysaccharide, a long chain called starch. So later on, when the plant needs energy, it can actually break these bonds, called hydrolysis from our previous lecture. And so if you add water here, you can actually break the bonds. And now you have glucose molecules that can be easily accessed by cells for energy. So think of like monosaccharides like glucose as kind of like your like quick snacks, like you're ready to eat. Think about these large molecules as like energy storage. So got like a lot of, let's say you have like a lot of snacks like on a, or think about it like, you know, those Lays that people get for graduation that have like candy on them sometimes. Think of starch as like a long lay that has a bunch of candy on it. So maybe you have it over your neck and you can eat it for a long time. So you can store store your sugar for a long time. Whereas if you just had one piece of candy, you eat that right away and it's gone. That's like starch. Starch is a long chain of sugar. Okay, so the first one. Starch was in plants. We eat starch though, and we can also get energy by breaking those bonds by doing hydrolysis to those bonds and getting energy. Okay, but in our cells, when we eat food, we eat sucrose, we eat fructose, we eat maltose, we eat lactose. What our body does is it digests those molecules by breaking them apart and then reshaping them to store them in our bodies if we don't need energy right away. And what we'll build is something called glycogen. So, glycogen is a bunch of these monomers strung together, saving energy for later. And we store glycogen in our liver. So when our liver is fully saturated with glycogen, if we have too much glycogen in our liver, we've eaten too much, then our body starts taking the excess glucose molecules and forming them, breaking them apart and forming them into fat molecules. So that's why you'll gain fat if you're eating more than enough glucose or sucrose or any type of carbohydrate, because your liver has too much. It doesn't need to store anymore, so it will start turning those molecules into fat. Okay, so just kind of a fun fact. Scientists created this thing called the glycemic index, and it's basically a ranking of how fast, if you eat a carbohydrate, how fast it gets broken up into smaller pieces, into smaller monosaccharides, which can eventually get into your blood. So in your blood, you can only fit like really, really tiny monosaccharides. You can't fit these long starch molecules. So how long does it take your body to break these down? It's called your glycemic index. So take a look at these options: rice, apples, rice cakes, cornflakes, bagels, peanut M&Ms. Which one do you think will get into your blood the fastest? Which one do you think your body can digest and break down the fastest? Take a guess. And let's take a look. So the food with the highest glycemic index is cornflakes. So that will be the one to get into your blood the fastest, as opposed to the other, like, let's see, peanut M&Ms, apples, less fast. Now let's talk about structure. So we talked about energy storage. So starch and starch and glycogen in plants. Plants store their energy in starch. In humans, we store our excess carbohydrates in glycogen. But let's talk about cellulose in plants. So cellulose is a polysaccharide. It's a long chain of sugars, and they're kind of in these rows. So you have one long chain here, one long chain here, and they're really, really, really strong. They form really, really strong connections, and that's because if you look between them, we've got these hydrogen bonds. So hydrogen bonds aren't super strong, but enough of them together creates a really strong structure. And this cellulose is actually the main component of cell walls. So if you were to look at a cell wall in plant cells, humans, we don't have cell walls, we only have cell membranes, but plants have these really rigid, strong cell walls made up of cellulose. And cellulose is the most abundant organic compound on Earth. Next, chitin. Chitin is found in the exoskeletons of some animals and also in the cells of fungus. So fungus is not a plant. It's made up of eukaryotic cells that are more like our cells than plant cells, but they're made up of chitin. So that gives the cells its structure. Okay, so just to review, polysaccharides are long polymers. They're long chains of smaller sugar molecules. It costs only a little energy to build. So remember we talked about how we eat kind of simpler sugars. We eat disaccharides. We eat sucrose, which is just two small monosaccharides together. And we have the ability really quickly to take small sugars and chain them up to store energy for a long time. So the main functions of polysaccharides are to store energy. So in plants, we store energy through starch. In animals, we create glycogen molecules to store energy for the long term. And then they also help us build our structure. So in cellulose in plants and in chitin in arthropods and fungi, creating the cell walls. Okay, finally, lipids are next. Not finally, there's more. Lipids are our next macromolecule. And when you think about lipids, you think about like fatty foods. Another word for lipid is fats. And we're going to talk more about their structure. All right, so the next macromolecule we're going to discuss are lipids. Lipids are, another word for them is fat, and they're composed of carbon, hydrogen, and oxygen, long chains. If you really look at the structure up close, if you were to differentiate a lipid from like a nucleic acid or a protein or a carbohydrate, you'd notice that there's just like really long carbon, hydrogen, hydrocarbon chains. There are different types of lipids. So one type are fats, that's a type of lipid. Another type is called a phospholipid. Another type is called a steroid. So in this video, in this section of the video, we're going to be looking at the structure of each of these fats, phospholipids, and steroids, the structure, and then their function. What is their job in the body? Now, lipids don't form polymers. So with our other macromolecules, we talk about the monomer version, the building blocks, and then we talk about how those monomers are chained together to form polymers. Lipids don't form polymers, but they do have like smaller pieces that they're made up of. So I'll kind of walk you through that. So lipids, they are bigger molecules and they're made up of smaller subunits, but not like repeating units like you would see. Proteins are just a repeating chain of amino acid monomers, or carbohydrates are a repeating chain of monosaccharides, or nucleic acids are a repeating chain of nucleotides. Lipids aren't repeating chains, but they are big molecules made up of smaller pieces, like I just said. Okay, so let's look at fat. So let's look at the structure of a fat molecule. You'll notice that there are two main pieces. On the left-hand side, this subunit here is called glycerol, and it's a 3-carbon alcohol. Alcohol, these OH group, that's a functional group called alcohol. And remember, anytime you see a molecule named with OL at the end, that is a name of an alcohol. So we have a glycerol molecule, 3 carbons with 3 OH groups here, 3 alcohol groups, and it's connected to something called a fatty acid. Kind of a funny word. So notice here we have a carboxyl group on the end. Carboxyl group is a carbon double bonded to oxygen, bonded to an OH group with a long hydrocarbon chain called a fatty acid. Now, if you were to just take a fatty acid on its own and you wanted to bond it to this glycerol molecule to create an entire fat, remember, how do you join two smaller molecules or monomers? You have to remove something. And so in this case, you're going to remove a hydrogen from the glycerol molecule and an OH group from the fatty acid. And now you've dehydrated. Dehydration synthesis. Dehydration means to take away water. Synthesis means to put together. So this carbon here will be covalently bonded to this carbon here, or to this oxygen here. And now you have attached them. So that is the structure of that. Now, fatty acids bind to glycerol molecules, but notice here that we have 3 fatty acid chains bonded to 1 glycerol molecule. And why is that? Well, notice here that there are 3 OH groups on the end, and so you can actually do dehydration synthesis 3 times to attach 3 whole chains of fatty acids on the end, and that will create a fat molecule. Now, another word for a fat molecule, and this is really important, is called a triglyceride. This triacylglycerol, I call it a triglyceride. I don't know why in this picture. Maybe it's a specific type, but it's called a triglyceride. And why is that? Well, tri means 3, 1, 2, 3, and they're all connected to a glycerol. And that happens through dehydration synthesis. So this glycerol head with the 3 tails, that is a fat molecule. All right. Now, technically, this bond here is called a ester linkage. It's a covalent bond between the oxygen and the carbon. That's not as important for you to know, but just so you know there. Yeah, so here, triacylglycerol. I don't say it that way. I say triglyceride. That's the way I say it. Okay. So fats, are they polar or are they nonpolar? Well, let's take a look at this molecule. Remember, polar means the charge is evenly distributed throughout the molecule. And you only really find polar molecules when you have electronegative atoms. So notice here, we do have oxygen. So you might think that this might be a little bit polar. That might have more electron density near the oxygen. But because this molecule is mostly made up of hydrocarbon chains are nonpolar. There's not really a positive or negative side. So we call fats nonpolar. So its polarity, the polarity of a molecule, will affect the way that it interacts in water. So if you put this fat molecule in water, what would happen? Would it be hydrophilic? Would it like water? Or would it be hydrophobic? Now remember, water is a polar molecule, meaning it has a slightly positive and a slightly negative end. And so if you were to put this in water, This is a nonpolar molecule. It doesn't have a slightly positive or a slightly negative end. So would it be attracted to the water? Would it be hydrophilic? Or would it not be attracted to the water? Would it be hydrophobic? Take a moment, think about it. It would be hydrophobic. Why is that? Well, this molecule, fat molecule, has its charge evenly distributed. It's basically neutral. And so if you put that in water, it will want to escape away from water. That's why fats and water don't mix. If you put oil and water, they don't mix together because fat is hydrophobic, afraid of water. Now, we talked about its structure. Now, what's its function? What do we do? What do fats do in our body? Well, the first main function of fats is they store energy. This hydrocarbon chain is packed with energy, and that gives your body energy for later. It's 2 times the energy of carbohydrates. So if you eat fatty foods, it gives you more energy quicker. It also cushions your organs, so there's a layer of fat around your organs, and that helps you helps protect them. And it also insulates your body, which means it holds in heat, like whale blubber. Keeps the whale warm. Okay, now there's different types of fats depending on how this hydrocarbon chain is oriented. So notice here that on the top we have just a long hydrocarbon chain, bunch of carbons, hydrogens, and notice here on the bottom it's a little bit bent. Now what is causing this bend? Well, we see here that this carbon is double bonded to another carbon. And when we actually look at the molecule physically, that molecule will be a little bit bent. So a saturated fat is a molecule that it's completely saturated with carbon. So what that means is every single carbon is fully surrounded by hydrogens. But if we look down here, this one is unsaturated. Unsaturated means it's not fully surrounded by hydrogen. Saturate means it's like full of, and this one's not, it's unsaturated. So there's not fully surrounded by hydrogens. So it's called unsaturated. Now, saturated fats are found in animals naturally, and they're solid at room temperatures, whereas unsaturated fats are found in plants and they are liquid at room temperature. And so we're going to look at what these molecules look like for real. So here's a saturated fat, here's our glycerol, and here's our fatty acid chains, so triglyceride. Here's also a triglyceride, but notice that they're a little bit bent. And so what does this mean for the fat? What does it mean for its structure and function? Well, if you took this fat molecule and then stacked up the same exact one on top and on top and on top, it would be like a really rigid structure. Whereas on this unsaturated fat, there's a lot of space in between. So if you were to stack up a bunch of unsaturated fats on top of each other, they wouldn't really be really tightly packed in. There'd be a lot of space in between. And so we'll talk in a moment what that means about its structure and its function. So saturated fats, just to review, you just have a long chain of hydrocarbons, and there's no double bonds. They're just a long straight chain. And these are found in animals. Now, because these hydrocarbon chains can really easily stack on top of each other without extra space in between, it makes them solid at room temperature. So if you took a bunch of fat molecules, unsaturated or saturated fat molecules, and you stacked them on top, you would make a solid. And so some examples of saturated fats are butters. Butters at room temperature is solid, and that's because it's made from animal fat, and it's unsaturated fat, saturated fat, which means the molecules are stacked on top really tightly. Now, saturated fat can contribute to cardiovascular disease, atherosclerosis. I'll show you on the next slide. So if you eat too much fat, then what will happen? Well, if we look here, here's an artery. Your artery is surrounded by some smooth muscle. And here's your red blood cells and white blood cells. But if you have too much fat, they can actually build up in your arteries and block the blood flow. And that could lead to blood clots and maybe even heart attacks if you have a buildup of too much saturated fat. And why is that? Well, it's solid at room temperature, and so it will just kind of like, it will take up a lot of space in your arteries. So then as opposed to unsaturated fats, so remember, unsaturated fats have that double bond, which makes them not fully saturated with hydrogens. We find this in plants and fish. Vegetable oils. So if you think about vegetable oil, that's made from unsaturated fat from plants, from vegetables. And at room temperature, vegetable oil is a liquid. So the kinks made by the double bond prevent the molecules from tightly packing together. So the straight saturated fats can stack on top, but because of that little kink, if you stacked them all up, they wouldn't be able to really form a solid. It would actually allow the molecules to kind kind of move past each other more easily, forming a liquid. Okay, so we just finished talking about fats. The next type of lipid we're going to talk about are phospholipids. Their structure is kind of similar, but a little bit different, and they also have a very different function in the body. So the phospholipid, it's made up of our glycerol. So down here, we have our glycerol up here. But notice that there are only two glycerols right here. There are only two parts of the glycerol molecule. It's made up of two fatty acids. So here's one saturated fatty acid chain. Here's an unsaturated fatty acid, because you see there, that's that double bond. Dehydration synthesis connects them to the glycerol, so two here. And then we have a phosphate group on top. That's a functional group. Phosphorus, double bond to oxygen, 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 oxygen. And these oxygens are negatively charged. And that's going to be really important in a moment. Other small molecules can also be attached to phospholipids called ATP. We'll talk more about that later. Okay, so let's think about this molecule. Is it hydrophobic or is it hydrophilic? Is it a polar molecule or is it a nonpolar molecule? Well, if we look at the fatty acid tails down here, it's just a bunch of hydrocarbons. This end of the molecule is hydrophobic because the charge is spread neutrally. But if we looked at the head up here, the phosphate group on top, the PO4, this piece is hydrophilic. It likes water. Well, why is that? Remember, water is polar, has slightly positive, slightly negative end, and that positive end, the hydrogen end, will be attracted to the negative oxygen. So this is actually a polar molecule, partially a polar molecule. We'll talk about, there's a word for that. So it's not totally polar, even though it has this kind of negative end. We call it amphipathic. And what that means is there is a hydrophobic end, and then we have a hydrophilic end. So this part likes water, this part does not like water. So it kind of has a dual personality. Can't decide which side. It can't decide if it likes or if it doesn't like water. One side does, one side doesn't. So their interaction with water is very important and complex. So if you put this molecule in water, I want you to make a prediction. What would it do if you put it in water? Which side would be attracted to water? Which side wouldn't? How would it move around? Now, why are phospholipids important? Well, let's take a look. So phospholipids, where are they found in our bodies? What's their function in our bodies? Well, they're actually found in your cell membrane. So the surrounding membrane of all of your cells, all cells, actually, every single cell in the world is surrounded by a cell membrane. And if we look here really close, let's look at this picture right here. Notice that here's the head. It's kind of hard to see. And then here's the two tails. So here's the head, the phosphate group, and then the fatty acid tails. Here's the here's the tails, head, tails. So we have a bunch, 1, 2, 3, 4, 5, 6, 7, 8, 9 phospholipids on top. Now, cells, on the outside of a cell and the inside of a cell is water. And so if you stuck a phospholipid in water, what would it do? How would it behave? Well, remember, the phosphate group has a negative charge, and so it's going to be hydrophilic. It likes water. So it's going to face the water. Whereas its hydrophobic tail, remember this is just a long chain of hydrocarbons, is going to face away from the water. And your cell membrane has two layers. So this top layer will have, if this is outside of the cell up here, this is the water surrounding the cell, all the hydrophilic heads will face the water, all the hydrophobic tails will face away. And then it's double layered. So here's the inside of the cell, which has some water in it as well. And here again, we have the phosphate groups, the head facing inwards because it likes water, hydrophilic. The hydrophobic ends will face away from the water. They will try to escape the water. So again, we kind of see a picture here. Here's one phospholipid, 2, 3, 4. Notice that the hydrophilic heads, the water-loving heads, face the water. The hydrophobic tails face inward. And this creates a really strong barrier where water can't really get in or out super easily. So it creates this membrane surrounding all of your cells. So again, if you put a phospholipid in water, its head, each individual head is hydrophilic. Remember, why is it hydrophilic? Well, because it's charged and it will like water, it will like bonding with water. Whereas the tail is hydrophobic and so it will hide away. So if you just put a bunch in water, they would aggregate or they would come together into this thing called a micelle or a liposome. It's just a bunch of phospholipids arranged in a circle because the insides are all trying to escape the water. The outsides are trying to go towards the water. And scientists think this might have been how cells were first formed. A bunch of fats, a bunch of phospholipids put in water. Because of their chemistry, because of their amphiphilic nature, part of it likes water, part of it doesn't like water, and so it forms these little micelles, it's called. Micelles are just a bunch of phospholipids in a circle. Okay, so again, we talked a little bit about phospholipids. Their function is they form the cell membrane, and we talked about how the cell membrane has two layers. We call it bilayer. Bi means two, so there's two layers, the top layer, the bottom layer. Remember, the hydrophilic heads are on the outside. They love water because they have that phosphate group that's charged. The hydrophobic tails are on the inside. They're escaping water, and this forms a barrier between the cell and the external environment. So this keeps all of the stuff inside the cell from escaping and things from outside the cell from coming in. So it kind of is like the guard or the wall of the cell. Now, your cell membrane also is composed not just of phospholipids. There's also proteins embedded in your cell membrane. There are carbohydrates, but we'll talk about that more later. Okay, so we talked about fats, we talked about phospholipids, and the last type of lipid we're going to talk about are steroids. So some examples of steroids in the body are cholesterol and sex hormones. These are examples of steroids. What is their structure? So there are 4 fused carbon rings. So here's 1, 2, 3, 4 rings. And they're attached to different functional groups on this end. So if you take this basic structure down here, but you kind of replace this top with different pieces, different functional groups, then you're changing the identity of the steroid. So for example, one type of steroid is cholesterol. Cholesterol is found in animal cell membranes. So if you look back here, the cell membrane actually has cholesterol embedded in. We'll see another picture of that in a moment. They're also called the precursor of other steroids. So this is kind of like the basic building block of other steroids, like sex hormones. We'll talk about estrogen. We'll talk about some more sex hormones as well, testosterone, and how those are built from cholesterol. Cholesterol is like kind of like the OG sex hormone before the sex hormones are created. We'll talk about that in a second. Too much cholesterol could lead to cardiovascular disease because just like you see here, here's a normal artery, but if there's too much cholesterol in your arteries, it could actually block and build up in your arteries, just like we saw with fat. And that could lead to heart disease. Okay, so the structure of cholesterol, it's again 4 carbon rings and then this functional group on the end, and they can be embedded into cell membrane. So here's our phospholipid membrane, phospholipid bilayer, but cholesterol can be embedded in. And then here's some other pictures of different ways to depict cholesterol. Now, cholesterol is the basic building block of sex hormones. So we all have cholesterol in our bodies, but what happens is this functional group will be modified. So if this functional group changes to an OH group, we call it estradiol, and that's a female sex hormone. If this functional group up here is replaced with, oh, nope, I got that wrong. So this functional group will both be replaced with OH here, but notice down here, the OH group stays, and it's estradiol. But if this hydrogen leaves and oxygen forms a double bond with this carbon here, then we call that testosterone. So this is kind of like the basic version, and then you will modify this end and this end to create different types of sex hormones. So even just one small little atom, the removal of this hydrogen makes testosterone. And that one difference is the difference between the development of male sex characteristics and female sex characteristics. All right, so just to review lipids, 3 types: phospholipids. This is the structure. There's 2 fatty acid chains collected to a glycerol and a phosphate group at the top. These are amphiphilic, meaning this is the hydrophilic side, the hydrophobic side, and these are found in cell membranes. Triglycerides, a glycerol bonded to 3 fatty acid chains, and these are found in animals and in plants. And we talked a little bit about saturated fats and unsaturated fats here. Then we have steroids. Steroids are hormones. They're also cholesterol. They help build the structure of your cell membranes. And that's a little review. All right, so the next type of organic molecule or macromolecule is nucleic acids. Nucleic acids, when you think of them, think DNA, think RNA, think ATP. So you can see like the structure of the nucleic acid here. We're going to break down what is a monomer of a nucleic acid, what are the bricks that build up a nucleic acid, and then what does the polymer look like. So we're looking here, this is actually the polymer, this is like the larger chain. But if we zoom in to this DNA molecule, we'd find individual building blocks or bricks that make up this larger molecule. So first, let's talk about what a nucleic acid is and its main function. So when we're talking about macromolecules, we want to think about its function in biological systems and also its structure. Its structure, what atoms make it up, what kind of bonds, what kind of monomers, what kind of polymers, how it gets formed. But now the function of a nucleic acid is to store and transmit hereditary information. So when you think nucleic acids, think it stores hereditary information or it stores your genes. So for example, we have RNA, which stands for ribonucleic acid. So the R stands for ribo, the N stands for nucleic, and the A is the A in acid. And we're going to look at a second at the structure of RNA, and we're going to find that each of these components of this word, ribonucleic and acid, are going to be seen in the actual shape of the molecule. DNA, deoxyribonucleic acid, is another type of nucleic acid, and its structure. So it's built up of monomers called nucleotides. So one brick is called a nucleotide. And we're going to take a look at what happens if you put multiple of those in a chain. So here's our brick. Here's the building block of a nucleic acid. Now, you'll notice here that this picture, this is one building block. This is one unit of a nucleic acid. But let's compare that to our other picture of DNA. So where do you see that in this? It doesn't look anything alike, right? And that's because it's kind of hard to depict the structure, like the monomer of a nucleic acid in this picture, because it's so detailed and complex. But if we really were to zoom in closely, we see this. So let me break it down for you. This right here is a monomer. If you put multiple of these together, it makes a long chain called a nucleic acid. So each of these, this is called a nucleotide, is composed of 3 items. The first one is a nitrogenous base. Nitrogenous means there's nitrogen in it. So if we look here, this shape here has a bunch of carbons. Each of these vertices represents a carbon, but scientists get lazy. They don't want to draw in all the carbons and the hydrogens, but there's nitrogen in here and it's carbon. And we call this a base. Now it's connected through a covalent bond here to a sugar. Now this sugar has 5 carbons in it. And again, scientists get lazy, but right here there's a carbon. There's a carbon, carbon, carbon. Even right here in this little elbow, that's a carbon as well. And then it has an oxygen in the center. We call this a sugar, and pentose, 5 carbons. And then on the end, the last part of a nucleotide is the phosphate group. So if you were to zoom into this little P here, you would find a phosphorus double bonded to oxygen, single bonded to other oxygen atoms, and that makes a phosphate group. Now, you'll notice here that each of these carbons has a label, a number. And that's because when we're looking at these carbon rings, these sugars, we actually need to count the carbons. It's going to matter later. So the way we count a carbon, the way we count a sugar, is we always start on the right-hand side where it's connected to the base. So we think of the first carbon as like the thumb. So it's like the sticking up and it's connected to that nitrogenous base. Now you don't want to go in this direction because then you're going to hit an oxygen. So this is the first carbon. Down here, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Now, later on, we're going to learn a little bit more about why this numbering of the carbon matters. But I want you to imagine like you were standing right here and looking like straight on with the nucleotide. This would be called the 3 prime. Prime is that little dash there. This is the 3 prime end down below. If you were standing right here, you would call that the 5 prime end of the nucleotide. Now, why don't we talk about the 1 prime or the 2 prime? Well, later we'll see that this part of the molecule actually is going to be in the inside of a DNA or RNA molecule. This part will be on the outside. And so we really typically speak of the 5 prime end and the 3 prime end, and I'll show you later what that really means. All right, so Something that you'll need to know later, though, we'll talk about this again, is that if you were to join this monomer with another monomer to create a polymer, you would join at the 3' end. So whenever we're talking about polymers, we think about the directionality, like where do new monomers add on to make a bigger polymer? We talked about that when we talked about proteins and how amino acids are chained up together. And so on N terminus versus C terminus. So go back and look at the protein video if you need or for a refresher on that. But in this case, if we were going to add a new monomer, we're always going to add to the 3' end of the nucleotide. Okay, so let's take a look here again just to review. So we looked at one nucleotide. Here's another picture, kind of a different artistic depiction. Oops, let me go back. Okay, so I'm trying to make myself a little smaller here. There we go. Okay, so once again, let's review. So here's our nitrogenous base. So this is made up of a bunch of carbons and nitrogen bonded to a pentose sugar. So carbon 1, 2, 3, 4, and 5 bonded to a phosphorus. And this is one monomer. But if you were to take this and copy, paste, copy, paste, copy, paste, here's number 1, number 2, number 3, and number 4. So you're copying and pasting a bunch together. And so again, 3 parts. We have our base, our sugar, our phosphate. Our base, our sugar, our phosphate. Our base, our sugar, our phosphate. Base, sugar, phosphate. So now we have a polymer. A polymer is a number of repeating monomers that all have each of these elements. Okay, now something you'll notice actually is that if we look at these bases here, they have a little bit of a different shape. This one has 2 carbon rings, this one has 1, 1, and 2. And that's because you can actually insert 5 different types of rings on a nucleotide. So let's take a look at some examples. So here we have a nitrogenous base called cytosine. And it looks like this. It has some carbons and then a nitrogen. That's why it's a nitrogenous base. Here's thymine, so a bunch of carbons. And they're pretty similar, but they have noticed just like slight differences, like the placement. Like this one has 2 double bond oxygens, this one has 1. You will not need to know that, but just notice that their structure is a little bit different. It makes them different molecules. Here's uracil, and then we have adenine and guanine. So notice that guanine have 2 rings, and adenine has 2 rings, and guanine also has 2 rings. So if we look here, these 2 rings are kind of represented here on our polymer. Now, we call nitrogenous bases that have 1 ring a special name. We call them pyrimidines. I'll say that again. Maybe say it to yourself. It's kind of a fun word. Pyrimidine. Pyrimidine. So pyrimidine is a base that has 1 ring. And then purines on the right, they have 2 rings. And so again, here we have purine, pyrimidine, and purine. So think of a way to remember that. Think of a way to remember that purines are 2 rings and pyrimidines are 1 ring. Think of some way to jog your memory. All right, so just like we talked about, there's different types of nitrogenous bases. So purines, 2 rings, pyrimidines, 3 rings. And we call each of them a different name. So cytosine, we can represent with the letter C, thymine, T, uracil, a U, adenine, an A, and guanine, G. So C, T, U, A, and G. Think back to your biology class. Did you learn about C's and T's and A's and G's and U's related to DNA? Well, here's that again. All right, so how do we take a monomer? How do we take a nucleotide? Remember, a nucleotide is just one of these. And how do we build an entire polymer? Well, we kind of just looked at it, but you take one, and here we have the 5-carbon sugar at the 3' end down here. How do you know it's 3'? Well, remember, we start counting here. This is like the elbow or the thumb of the sugar that's connected to the base. So 1, 2, 3. So that's a 3', 4, and 5. And I think of the 5' as kind of like the chimney. Like there's this little phosphorus or phosphate group that's sticking out. So the 3' end is connected to a new phosphate group, which is connected to a base, a sugar and a base. And then again here, this 3', how do we know that's 3'? Well, this is carbon 1, carbon 2, carbon 3, covalently bonded to our next one, and it keeps going. So that creates a longer chain. Now, one thing I want to note is that the sugar actually has a special name. So in DNA, we call it deoxyribose sugar. And we'll kind of talk about that in a moment, but think about when we name this sugar, it's called deoxyribose. However, when we're talking about RNA, the shape of this sugar right here is actually a little bit different. We call it just ribose. Now, why is that? Well, let's look closely at the chemistry of these molecules. So deoxyribose, D means not. And so deoxy means like less or one less oxygen, no, not oxygen. So do you notice in this picture a lack of oxygen compared to this ribose over here? Take a moment, look closely at it. Do you see less oxygen on this side? Deoxy, less oxygen. Well, yeah, notice here on the second carbon, it's connected to just a hydrogen, whereas on the ribose, we've got our first carbon, second carbon, and then an OH. So ribose is a 5-carbon sugar that has an OH. Deoxy has one less oxygen, and that difference is the difference between DNA and RNA. So you'll see a ribose sugar. That's why it's called ribonucleic acid. This is a sugar that's placed here in RNA, and then in DNA, it's called deoxyribonucleic acid because it has one less oxygen. So when you're building up a DNA molecule, you'll have deoxyribose in that sugar spot. So all of these in DNA will be deoxyribose. Whereas if you're building RNA, all of these sugars would just be called ribose. All right, so when we're talking about this polymer here, this long chain of monomers, remember we have the phosphate, the sugar, and the base. Here is our purine, here's our pyrimidine. Notice that there is a covalent bond between our sugar and our phosphorus group. It's called a phosphodiester bond, but that's just a fancy word for the special covalent bond that's found between the sugar and the phosphate group. When new nucleotides are added on, they're always added on to the end of the 3' end of the sugar. And the polymer only grows in one direction. So the new chain will never grow on this phosphate group. The new chain will always grow down here. A new monomer can be added to the 3' end of this sugar down here. Okay, so then that's how you build a polymer. Okay. Oh, and then one thing to note, we call the outside piece, so the phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. We call this a sugar phosphate backbone because we'll see in a moment that it's going to be on the outside of a DNA molecule, and then these bases are on the inside. We'll see that in a second. So this is the backbone. It's like the outside. It's holding everything together. All right. Now, when we're making DNA, it gets a little bit more complex. So remember how we made this long chain, this polymer? Well, in DNA, there's actually two of these side by side. So 2 strands. So if we look here, let's review. So here's our phosphate, here's our sugar, here's our base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. So this right here is one strand. Let me see if I can use my pen. So this is one strand of DNA. And this side is another strand. There's 2 strands running parallel to each other. So notice that this strand is bonded, the bases in the center, the nitrogenous bases are bonded with what kind of bond? Notice it's a hydrogen bond. So because of the atoms that are making up these molecules here, they are interacting through hydrogen bonding. And those are weak bonds. So these bonds can easily be broken and formed. So what that means is those 2 strands can be ripped apart. And that's going to be really important when we talk about DNA replication and protein synthesis, making proteins. Remember that the function of DNA is to store genetic information. And we'll learn in a moment that these bases, the order of the bases are like a code. So we have T, G, C, A, and that code needs to be read by your cells. And so in order for your cells to access that code, you actually need to rip apart these hydrogen bonds eventually when you're making proteins. So that's why those hydrogen bonds are important. If DNA was bonded, each strand was bonded with covalent bonds, it would take so much energy to rip those covalent bonds that DNA wouldn't rip apart. So that hydrogen bonding is really important. Notice that here we have a pyrimidine, 1 ring for the base. Here we have 2 rings. And so notice that Pyrimidines are always bonded to purines. So here we have a purine, pyrimidine. We have a pyrimidine, purine, purine, pyrimidine. Notice that T is always bonded to A. And whenever you have a T and an A bonded, there's always 2 hydrogen bonds. That's just kind of something you have to memorize. Think of a way to memorize that. And then another way that I remember that T and A bond together is they're kind of like the sticky, pointy ones. Like if you were to like touch a T or touch an A, it feels like they would like poke you. Whereas G and C always pair up together in DNA. So G guanine, C cytosine. And notice that G and C always pair up with 3 hydrogen bonds. And this is a little stronger. So the more hydrogen bonds you have, the stronger your DNA. So if you have more G's and C's in your DNA, it's a little harder to rip apart. Okay, I think we talked about that well enough. Okay, so why is hydrogen bonds important? Think to yourself, that's an important piece here. Okay, now this isn't as important, but some organisms have more G and C bonds than others, and so that makes the DNA even stronger. Now, in order to separate bonds, like when you're in the lab, sometimes scientists are taking DNA and they're separating it and trying to make new copies of DNA. They heat up the DNA, and the heating up actually can break these hydrogen bonds. And so, if you have more G's and C's, the G's and C's have stronger bonds, more hydrogen bonds, it's harder to separate the strands. And this is interesting too. Organisms that can survive at really high temperatures, they need to keep their DNA intact. So they have more G and C, more G's and C's in their DNA compared to A's and C's. So just a kind of interesting fun fact. And that keeps the molecule more stable. So if it is exposed to higher temperatures, it's harder to break those bonds. Higher temperatures typically break bonds because a bond needs an input of energy in order to break. And so heat supplies energy to break that bond. Okay, so let's review. So this picture, we're just going to kind of go in. We've talked about each piece, but let's go over the whole thing. So when you first look at this, it looks pretty confusing, but we've talked through it all. I'm going to repeat kind of everything we just said. So let's first find our monomer. So our monomer is the basic building block, and that would be one of these here. So we have our sugar, our phosphate, and our base. We have one ring here. So is that a purine or a pyrimidine? It's a pyrimidine, one ring pyrimidine. And notice here that it has, it's connected to our sugar. Here is carbon number 1. How do you know it's carbon number 1? Well, it's connected here. It's like the thumb to our base. And then if we continue on, carbon 2, carbon 3, there's our 3' end. And remember, our 3' end is always connected to our next monomer. Carbon 4, carbon 5, and then there's like a little chimney, the phosphate group. Okay, notice here that the purine and the pyrimidine, C's and G's, are connected by 3 hydrogen bonds here, whereas here we have a thymine and an adenine. T's and A's are connected by 2 hydrogen bonds, so that's something important to remember. Let's see, what else do I need to show you? Oh, let's take a look at the 5' and 3' end. So we call this end of the molecule the 5' end, and let's review why. Well, if we're counting our carbon, so this is our top carbon, or top sugar here, so this is where we start. So this is carbon 1, 2, 3, 4, and 5. So we call this the 5' end because notice that this ends on the 5 carbon. Whereas if we look down here, we call this a 3' end. Well, why is that? Well, let's find our carbon 1. So carbon 1 would be here because it's like the thumb, it's connected to our base. Carbon 2, carbon 3, and so that here, down here, is our 3' end because it ends on our 3rd carbon. Okay, so this is the directionality of the molecule, 5' to 3'. However, let's look at this strand on the right-hand side. Notice that at the top it says 3', and on the bottom it says 5'. So when we're talking about DNA strands, we talk about them running anti-parallel. So parallel means kind of side by side, and anti means opposite. So they're running in opposite directions, and you can even see here that the sugar is like flipped upside down compared to this. So let's review our 5' and 3'. Well, first let's find our carbon 1. So if we look at this sugar here, which one's our carbon 1? Now remember, carbon 1 is always connected to your base. So this is our first carbon, And then we can't go this way because that's an oxygen. So let's go here, carbon 2, carbon 3. So that's our 3' end. Carbon 4, carbon 5. And then our next nucleotide down here is always connected to our phosphorus. Okay. All right. So let's count this sugar over here. So here's our carbon 1 because it's connected to the base. Carbon 2, carbon 3, carbon 4, carbon 5. So we call that the 5' end. So they're running anti-parallel directions. So now we've looked at this entire piece of DNA, and this stretches will keep going hundreds of bases long. And this is just an example of a couple of nucleotides pairing up. So nucleotide number 1, nucleotide number 2 pairing up, nucleotide number 3, and nucleotide number 4. And then it will continue on a long chain to make a whole thing of DNA. All right. I'm going to figure out how to clear this. Fireworks. Woo. Oh, wow. That's fun, but that's not what I wanted. Eraser. Clear screen. Boop. Okay, there we go. All right. Fun things. Mouse. Okay. So we basically went over everything. Something to note is that oftentimes DNA will be depicted like this, which you're like, whoa, what is this? That looks very different than our previous few screens. That looks really different than this. Well, it's the same thing, it's just drawn differently. So right here, this backbone here, this ribbon, is the sugar phosphate backbone. So if scientists are lazy, they just draw this backbone. So basically, this piece right here, the phosphate sugar, has a backbone. So it just has this ribbon. And then in the center piece, these are the bases. So GC, AT, TA, GC, and you can see these hydrogen bonds between the bases. So the bases are facing on the inside of the molecule, and then the sugar phosphate backbone is kind of on the outside. And then it's called a double helix because the two strands are like a twisted ladder. If you took a ladder and you twisted it, it would create this helix shape, and that is a DNA molecule. All right, now don't forget, why is it important that there are hydrogen bonds between the bases? Think about that. All right, now DNA isn't the only type of nucleic acid, but the next type is very similar. So DNA is two chains, so two strands wound together, like we see here. So here's one chain, one backbone, bonded with another side. So two strands running anti-parallel, spiraled in a double helix. And this molecule was discovered by Watson and Crick and Rosalind Franklin and many others who contributed research to discovering this molecule. But this is very similar to another molecule called RNA, or ribonucleic acid, which is just a single nucleotide chain. So remember, nucleotides are the little building blocks that make up an entire molecule. And so RNA looks more like this. So let's look at it. So here's our backbone. Here's the ribbon backbone. And if we zoom in, we can see the phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. But think again, what is the name of this sugar? Is it deoxyribose or is it ribose? If you said ribose, you're correct. So this is a ribose sugar. That's one thing that makes RNA unique. The next piece is that it's single-stranded. So DNA is double-stranded. Here we don't have the bases connected to anything else. They're all alone. And the last piece that's different, notice we have guanine. So we have our purine here. We have adenine, our purine. We have cytosine, our pyrimidine. But now we have U, which stands for uracil. And so in RNA, instead of T's, we have U's. That's just something to memorize. This is an RNA molecule. So if we look closely, I talked a little bit about that sugar. So here is the sugar that's in DNA, the ribose sugar. And we have, again, notice that this second carbon, so carbon 1, carbon 2, is only connected to a hydrogen. So we call it deoxyribose, less than one oxygen. And then here we have a ribose. So carbon 2 has an OH. And this is the sugar that's found in RNA. Now there's another type of nucleic acid called ATP. This is not a molecule that stores genetic information, but it does store energy. This is a main energy currency for our cells. Hopefully you've heard of ATP before in your biology class. And notice that it looks very similar to DNA and RNA, where it has this base here, this nitrogenous base, adenine. That's why it's called adenosine, because it's made up of a base called adenine. Then it's connected to a sugar, connected to ribose, just like RNA. And then it's connected to phosphate groups. But notice in our DNA and our RNA, just connected to one phosphate group. But here, ATP is called triphosphate. Now that root tri means what? 3, right? 3. So 1, 2, 3 phosphates bonded together. So it's like a nucleotide that's a little bit modified. So it's like DNA and RNA, except now you have more than one phosphate group. So if you look here, here's our phosphate, here's our ribose, here's our base. But if you add another phosphate and another phosphate, whoop, that's ATP. Now we'll learn later there's even another molecule called ADP, adenosine diphosphate. Di means 2. And so if you just have 2 phosphate groups here and one's gone, that's ADP. Now, the addition of a phosphate group or the breaking of a phosphate group gives your cells energy. And so we'll learn more about that when we talk about photosynthesis and cellular respiration. But this is the reason why you're alive. This molecule is the reason why you're alive. It's the energy currency for your cell. So we'll talk more about that later. Okay, so last thing, what does your DNA do? Well, it's a series of bases. It's a bunch of A's, T's, C's, and G's that encodes information like a book. So if you're reading a book, you look at the words in the book and you can kind of decode it. It's a message that you can understand. In the same way, the order of A's, T's, C's, and G's in my DNA compared to your DNA is different. And because our DNA, our code is different, we have different instructions for how to build the way we look. And that's actually passed from parent to offspring. So my parents passed DNA to me, and their DNA is now partially what I made, what my DNA looks like. It's a mix of both my parents, and their DNA is a mix of their parents. And so DNA is passed down from parents to offspring, and that's why we look similar to our parents sometimes. Okay, and then if we look even closely here, here's our DNA. And if we coil it up really, really, really tightly, it makes up chromosomes. And then if we put those chromosomes in a cell, they're found in the nucleus. And in humans, we have 46 chromosomes, or 23 pairs, and they're made up of DNA. All right. Congratulations, you made it to the end of the video. If you've made it here, that means that you've learned a little bit about the 4 major biomolecules, macromolecules, organic molecules, and hopefully you've been filling along this chart as you go. But let's just go over it and make sure you have this in your notes. So carbs, the monomer sugars are the monomer of carbs. An example of sugar is glucose, fructose, and galactose, and this is the basic structure. Remember, if you put 2 sugars together, they form disaccharides. Just a simple sugar is called a monosaccharide. So maltose is a disaccharide, glucose and glucose. Sucrose is a disaccharide, glucose plus fructose, and lactose is a disaccharide, glucose plus galactose. Now, if you take a monosaccharide and put it in a long, long, long chain called a polysaccharide, that makes a polymer. Some examples of polysaccharides are starch, glycogen, cellulose, and chitin. And here we can see the functions of each in living things. Second, macromolecule lipids. There are 3 main types: fats. I don't know where my phospholipids are. Fats, phospholipids, and steroids. And the structure of fats are fatty acid chains, so these long chains, these hydrophobic chains, with the glycerol head. And then steroids are a bunch of 4 carbon rings, and then if you change the ends of the steroids, it changes the identity of it, so making testosterone or estradiol, which are sex hormones. Steroids are another word for hormones. And then if you were to join, they don't really have a polymer of lipids, but fats and oils are examples of lipids. And here's the function: phospholipids, steroids, and waxes. Even we didn't talk about waxes in the video, but that's another type of fat or lipid. Proteins made up of monomers called amino acids. And remember, we have the central carbon bonded to a variable R group. There's 20 different types you could plug in here. Some are polar, some are nonpolar, some are acidic, some are basic, some are hydrophobic, some are hydrophilic. And then we always have our N terminus, or our amino group. This is a amino functional group. And then we have our carboxyl group. Now, we didn't talk about this as much in the video earlier, but this is the amino side and this is the acid side. Why is this an acid? Well, this OH can actually release or dissociate or release its hydrogen. And if a solution has more hydrogen atoms, it is more acidic. So this side will often donate its hydrogen to the solution if you put it in water, making the more acidic. That's why it's called an amino acid, because this is the amino side, this is the acid side. Remember, these amino acids form long chains to form total proteins. And go back, the protein video is so important when we talked about the structure, the primary structure, secondary, tertiary, quaternary structure. And don't forget to think about how if you change one piece of each primary, secondary, tertiary, quaternary, how that would affect the shape of the protein and the function of the protein. Some examples with functions: proteins make up muscles. They are used as messengers throughout the body. They fight. They're antibodies. We didn't talk about that earlier, but they fight off invaders. They're used as enzymes. They break down molecules. They're found in your hair and your nails, and they are actually in membranes. They're found in your membranes as channels to let things come in and out of your cell membranes. And then finally, nucleic acids. The monomer is a nucleotide. So here's the phosphate group. Here's the sugar, pentose sugar, and here's the nitrogenous base. And we talked about 3 types of polymers. If you put these in a long chain and they were double-stranded, you'd find DNA. The function is genetic code. If you just had a single strand with a ribose sugar and then U instead of T, that's RNA. RNA is used to build proteins. We'll talk about that later. And then the last type of nucleotide is ATP. If you took this basic structure, but you added 2 more phosphate groups, that's adenosine triphosphate, and that gives your cells energy. So that's a little review. Keep this in your notes. This is super important.